Welcome to your first web lecture for B Core 12, Exploring Biology, and we're going to start off the semester with the topic of meiosis. So this is going to be similar to and follow up from the unit that you did on mitosis back in B Core 11. If you did not take B Core 11, you probably will want to go back and make sure that you've read that chapter on mitosis, as the mechanisms and the way things move, the way things happen, is going to be identical between mitosis and meiosis, but meiosis is going to have some important differences from mitosis. So why are we starting this whole semester with meiosis? So this first unit that we're going to tackle in this class is going to be a unit on evolution. So evolution is the driving force that results in organismal diversity. So before we go into examining the diversity of life on Earth, we're going to take a very close look at the process that results in that diversity. So this whole first section, this is a list of topics from your syllabus for the first part of the course. And this is all going to be leading up to this idea of organismal diversity. So what does meiosis have to do with evolution? So let's think back to that very first chapter that you read in B Core 11, where you got sort of the general overview of evolution by natural selection. And in that chapter, you saw that Darwin made a series of observations. So he observed that individuals in a population vary in their traits, and many of those traits seem to be heritable, so they're passed on from parent to offspring. He also observed that more offspring are produced than survive, so population sizes tend to be relatively stable. If all of those offspring survived, population sizes would increase out of control. Because of that, competition is inevitable. Not all of the offspring in any particular generation are going to survive to reproduce. And he also noticed that species are generally well fit to their environment. They have the tools they need to survive in whatever environment they're in. So from these observations, Darwin reasoned that individuals that happen to be best suited to their environment are more likely to be the winners in this competition for survival. They're more likely to survive and reproduce, and any traits that they have, any characteristics they have that help suit them to their environment is also going to increase their reproductive success. We can plan on them having more offspring than individuals that don't have that trait. So over time, more and more individuals in a population will have those advantageous traits because of the reproductive advantage inherent in having those traits. And so this is basically a description of evolution occurring as the unequal reproductive success of individuals in each generation that's going to cause a change over time in populations, not in individuals, but in populations. And this is the idea that the natural environment selects for the propagation of traits that are beneficial in that environment, and this is basically natural selection. So this is all a very nice and neat story. It is all held up to vast scrutiny in the scientific world since then. And Darwin really had nailed this idea very carefully with lots and lots of evidence that showed the soundness of this reasoning. But the one part of this that he really didn't nail is, although he observed that individuals in a population vary in their traits, and that offspring tend to resemble their parents, and so these traits can be heritable, he really did not know anything about the mechanisms by which traits are passed on from parent to offspring and what causes that variability in population. There was nothing known at the time about genes or genetics or DNA. The only thing Darwin had to go on were these observations of variation and heritability. So what we're going to start with is to think about what are the causes of that genetic variability in population that causes this variation in traits that is the raw material that natural selection selects from. So here's an example of the kind of thing Darwin observed, that offspring resemble their parents, but not exactly, and siblings resemble each other, but not exactly. So there's this variation in any sort of population, and this is the topic of heredity. This is what heredity is all about. So we now know that offspring acquire genes from parents by inheriting chromosomes. Chromosomes are the actual units that are passed from parent to offspring. So in a literal sense, children do not inherit particular physical traits from their parents. When somebody says, oh, you have your father's eyes or your mother's skin, that's not literally true. What you got from your parent are genes that control the building of those traits. So it's the genes that are actually inherited, and the unit that is passed from parent to offspring is the chromosome. So as we saw last semester, most DNA is packaged into these units called chromosomes, and a chromosome is one very long DNA molecule that will have a number of genes on it. So a gene's specific position along a chromosome is called its locus. So a gene has a particular location on a chromosome that's going to be consistent among all the members of that species. During sexual reproduction, reproductive cells called gametes unite to form a new individual, and this process is called fertilization. So gametes are called sperm and eggs in animals. These are the sex cells that are responsible for reproduction in sexually reproducing species. So for example, humans have 46 chromosomes in the nuclei of all of their somatic cells. Each cell has the same 46 chromosomes with all the same genes, all the same DNA, all the same genetic information. Every cell of the body except for gametes and their precursors. What happens in these gametes, these sex cells, is a process called meiosis. It's a nuclear division that leads to a halving of the chromosome number. So these gametes will have half the number of chromosomes that somatic cells possess. So at fertilization, when two of these gametes come together, they combine their half complement of chromosomes together to restore the full chromosome number. So in the case of humans, that would be 46. So of course, not all species reproduce sexually. Asexual reproduction exists in the world. There are many species that are capable of both asexual and sexual reproduction. And so let's take a look at the relative advantages and disadvantages of sexual and asexual reproduction. So in asexual reproduction, a single individual passes all of its genes to its offspring without the fusion of gametes. So this is basically going to happen by mitosis, producing an entire individual from one of the cells of its parents. So the full genome of the parents is passed on directly to its offspring. And so we've seen this in cell division, so unicellular organisms that just divide to reproduce is using a form of asexual reproduction in which the whole genome is passed on directly from the parent cell to the two daughter cells. This happens in many plants that can reproduce, often both sexually and asexually. So the example here you see is a strawberry plant that sends out these runners. This is a modified stem that then produces roots and creates another plant. If you were looking at a field of strawberries, you would say that these were all different plants, but basically these are the offspring of this parent plant that produced them asexually. When you take cuttings from a plant, that's basically asexual reproduction. So when a stem is able to produce new roots and create a new plant, and again, that whole genome is passed down intact. There are many animals that also can reproduce asexually in a number of ways. So this is an organism called a hydra, a relative of jellyfish, that can actually bud off new individuals from its stock. And this individual, once it's mature, will detach from the parent and become a new individual. And there are other animals such as the aphids that you see here, and even some vertebrates such as some lizards and amphibians can reproduce asexually through a process called parthenogenesis, where a female is able to produce offspring without any fertilization of eggs. So a clone is a group of genetically identical individuals from the same parent. So this implies that they have all of the same DNA and they're going to be genetically identical. In sexual reproduction, two parents give rise to offspring that have unique combinations of genes that were inherited from the two parents. So it's going to be some kind of combination of all of the characteristics of the two parents. So in sexually reproducing organisms, there's going to be this cycling between phases of gametes, fertilization, production of an adult, and then formation of gametes again. And we call this a life cycle or sexual life cycle. So a life cycle is the generation to generation sequence of stages in the reproductive history of an organism, from fusion of gametes, in other words, fertilization, to the formation of a zygote, to cell division by mitosis, which results in development into an adult. The adult then produces gametes, which then repeat this process. Meiosis and fertilization result in changes in the number of chromosomes per cell. So remember that each of these gametes has a half complement. When they come together, they have a full complement of chromosomes, and they're going to be cycling back and forth in every generation between these haploid cells and the 2N or diploid individuals, where N refers to the number of chromosomes. So how do we study and analyze chromosomes? So we find when we look at them microscopically that chromosomes, when they're condensed and ready for mitosis or meiosis, form distinct sizes and shapes. So what we find is that every species has a characteristic number of chromosomes. So in sexually reproducing species, there are sex chromosomes that are going to determine the sex of the individual. So in many animals, females have two X chromosomes, and males have an X and a Y chromosome. And so these chromosomes are actually distinct from each other. In most cases, the X chromosome is much larger than the Y chromosome, although it's not depicted in this illustration. And then there are other sets of chromosomes that have their own particular sizes and shapes. And these other chromosomes are known as autosomes. Those are the non-sex chromosomes. So for example, human somatic cells have 23 pairs of chromosomes. And we can visualize these 23 pairs of chromosomes through a visualization technique called a karyotype, which matches up the pairs of chromosomes based on size and shape and orders them from largest to smallest. And then we can number them. So here we see 22 pairs of autosomes and then a pair of sex chromosomes in this individual who happens to be male. So the two chromosomes in each pair are called homologous chromosomes or homologs, and we can pair up homologs based on the length, the shape, and also the way they take up certain stains while they're being processed. They're going to have a particular banding pattern and color pattern, and so they can be matched up using computer algorithms to show these homologous chromosomes, these ones that match. And each member of this homologous pair of chromosomes is going to have the same genes on it in the same locations. So it's going to have the same set of loci, the locations where particular genes are found, and they're going to have two copies of the same gene on each of these homologous chromosomes. So again, human females have homologous pairs of X chromosomes, and human males have one X and one Y. So here we see the large X chromosome, the smaller Y chromosome. So each of these pairs of homologous chromosomes represent the contribution of each parent. So each one of the pair is going to come from one parent. Say this one comes from the mother, and maybe this one comes from the father. So this is going to be the contribution of the two parents. Each one is going to contribute one chromosome in each set. So the 46 chromosomes in a human somatic cell are two sets of 23. One set of 23 came from the mother, and one set of 23 came from the father. And we call a cell with this complete complement of two sets of chromosomes, we call this cell a diploid cell. It's got two complete chromosome sets. A gamete with half of the number of chromosomes or only one chromosome set is known as a haploid cell. So for humans, the diploid number is 46. And we, again, refer to the number of chromosomes in a set as N, and this is going to have two N's. So let's just take another look at this terminology. Some of this will be familiar from your study of mitosis. So after the DNA has been duplicated in preparation for the division of the cell, what we end up with is a pair of sister chromatids. So these two that look like they're kind of squished together and very tightly adhered to each other are exact copies of the same chromosome. So these are known as sister chromatids. They are duplicate copies of one chromosome after DNA synthesis in preparation for division. In the case of this duplicated diploid cell, we see we have two copies of each chromosome. So these pairs are known as homologs. So we see three different pairs of homologs in these 6 chromosomes, these 6 duplicated chromosomes. In these homologous pairs, we can say that there are chromatids that are homologous, but they are not sister chromatids. So these are not exact copies of each other. Remember, one of these came from the mother, one of them came from the father. They're not exact copies, but they are homologous chromosomes. They have the same sets of genes on them, but these are two non-sister chromatids in a homologous pair if we look at these two that are next to each other. In summary, this cell that's preparing for division and has been replicated has twice the amount of DNA as a regular diploid cell. It is still considered diploid. It still has 2 homologs of each chromosome, 1, 2, 1, 2, so it has a haploid number of 3 and a diploid number of 6. So now let's take a look at the behavior of chromosome sets in the human life cycle. So again, fertilization is the union of gametes, in our case a sperm and an egg, and that fertilized egg is known as a zygote. So a zygote is diploid. It's a single cell that results from the fusion of the 2 gametes to form a diploid cell. It has 1 set of chromosomes from each parent. The sperm had a full set of chromosomes, the egg had a full set of chromosomes, and now it's going to form the diploid set with 2 copies of each chromosome. The zygote produces somatic cells by mitosis to allow that cell to grow and develop into an adult. And then at sexual maturity, the ovaries and testes in that adult human are going to produce haploid gametes with just 1 set of chromosomes in each gamete cell. Gametes are the only types of human cells produced by meiosis rather than mitosis. So a diploid cell can go through meiosis and produce haploid gametes. So meiosis results in 1 set of chromosomes in each gamete. Fertilization and meiosis alternate in sexual life cycles to maintain the chromosome numbers. So we've got a cutting in half and then a doubling up to maintain that constant chromosome number through the generations. And so this alternation of meiosis and fertilization is common to all organisms that reproduce sexually. But there's variations on this theme based on what part of the life cycle produces multicellular organisms. So the 3 main types of sexual life cycles differ in the timing of meiosis and fertilization, and whether diploid or haploid or both phases result in a multicellular organism. So depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. So in a typical animal life cycle, we call this a diploid life cycle. Gametes do not undergo mitosis. They do not divide and become a multicellular organism. All they do is combine with another single gamete in fertilization, and then mitosis occurs. In another kind of life cycle that's common in plants, both the haploid and the diploid phases result in multicellular organisms. So there's mitosis after fertilization to produce the adult plant. Meiosis occurs. Some of these cells form gametes. Some of them undergo mitosis in the haploid form to form a multicellular organism that we call a gametophyte. And then fertilization occurs using these single gamete cells to produce another multicellular organism in the diploid state known as a sporophyte. Don't worry too much about these details right now, but this is going to become incredibly important when we start to look at the evolution and diversification of plants. A third major form of life cycle is called a haploid life cycle, in which the diploid phase does not undergo mitosis, does not undergo cell division to form a multicellular organism, but the haploid phase can. And so this form of life cycle is found in most fungi and also some single-celled protists. So even though both diploid and haploid cells can undergo mitosis to form these different kinds of life cycles, the only kind of cell that can undergo meiosis to form these haploid gametes are diploid cells. So in all three of these different kinds of life cycles, the halving and doubling of chromosomes contributes to genetic variation in offspring, and we're going to start to examine how that happens as we go along here. So let's take a look at the details of how meiosis happens. So meiosis reduces the number of chromosome sets from diploid to haploid, from 2 sets to 1 set. So like mitosis, meiosis is preceded by the replication of chromosomes. So for each homologous pair, we're going to have a pair of sister chromatids. Meiosis is going to take place in 2 consecutive cell divisions, called meiosis 1, which is going to result in 2 daughter cells, in which the homologous chromosomes separate from each other, and meiosis 2, each of these is going to divide again to separate the 2 sister chromatids, resulting ultimately in 4 daughter cells rather than the 2 daughter cells that we see in mitosis. So 4 daughter cells, each with one of these 4 chromatids that we see in the replicated cell. And then each daughter cell has only half as many chromosomes as the parent cell. So remember, this is 2 chromosomes. They're each duplicated, but each of these is 1 chromosome, and each one of the daughter cells is going to have half as many chromosomes as the parent cell did. So now let's take a closer look at these stages. So as I mentioned before, the chromosomes duplicate before meiosis begins. So here we have the original cell, and all of that DNA is going to replicate in preparation for division to form 2 sister chromatids for each chromosome. So these resulting sister chromatids are going to be tightly adhered to each other. So they're going to be closely associated along their entire length, so this is why we show them kind of squished together. And this is called sister chromatid cohesion. They're stuck together, and then ultimately those chromatids are going to be sorted into 4 haploid daughter cells. So again, meiosis consists of 2 cell divisions. Meiosis 1 is where the 2 homologs of each chromosome pair separate into 2 daughter cells. Let's say the blue ones are the paternal chromosome set, the red ones are the maternal chromosome set. The 2 homologs in this first stage of meiosis are going to separate into separate cells. At this point, we go from diploid to haploid. Okay, so even though there are 2 sister chromatids, remember these are identical copies of each other, so this is the haploid chromosome number, even though there are 2 sister chromatids. So we've gone from diploid to haploid in this first division in meiosis. In meiosis 2, this is where the 2 sister chromatids of each chromosome separate into 2 daughter cells. So the 2 copies of the paternal cell are going to go into 2 separate gametes, and same with the 2 copies of the maternal chromosomes. And again, in the end, this results in 4 haploid cells. And the phases of meiosis during each cell division, during meiosis 1 and meiosis 2, are going to parallel the phases of mitosis that you learned last semester. So we're going to have prophase 1 in meiosis 1, in which the spindle microtubules assemble to form a spindle apparatus in exactly the same way that you saw in mitosis. The chromosomes are going to condense, having been replicated. They're going to condense down into that form in which you can visualize them microscopically. During metaphase 1, rather than all of the chromosomes lining up on this metaphase plate, this equator between the 2 spindle poles, the pairs of homologs are going to line up together. So both of the 2 homologous chromosomes are going to line up next to each other, attached to these kinetochore microtubules. One from each pole is going to attach to one of the 2 homologs. Anaphase is going to be exactly analogous to anaphase in mitosis. This is where the kinetochore microtubules are going to disassemble from the kinetochore end, and these tubules will shorten, pulling those chromosomes toward one end of the cell, separating them, and then finally telophase and cytokinesis is going to separate the two sets of chromosomes into new nuclei and complete the process of cytokinesis or cell division using a cleavage furrow in the case of animals and that mitotic plate in plant cells. So let's just recap some of the important features of that process. Remember, meiosis 1 is a reduction division. It's at this point that we go from diploid to haploid, reducing the chromosome number to the haploid number. In most plants and animals, a diploid cell produces 4 haploid daughter cells. But for meiosis 1, we're basically going to 2 haploid daughter cells, each of which contain a pair of sister chromatids for each chromosome. So remember, they're haploid, but they still retain these replicated chromosomes. So in order to get 1 copy in each of 4 daughter cells, they need to divide again. So in addition, they have a random assortment of maternal and paternal chromosomes. And this is going to be due to a couple of different processes, crossing over, which we'll look at in more detail in a moment, and also the random distribution of maternal and paternal homologs to daughter cells. So what we're looking at now is just a single chromosome, but if we had more different chromosomes in the cell, the paternal ones and the maternal ones can distribute at random into these 2 cells at the end of meiosis 1. We'll talk a little bit more about that a little bit later in the lecture. So meiosis 2, the second cell division, each of those 2 cells that were formed in meiosis 1 are going to go through this process of dividing again to form 4 cells. So this is represented by these 2 cells here. And exactly the same set of phases. We've got prophase 2, in which the mitotic spindle reforms and these centrosomes move to the 2 opposite poles of the cell. In metaphase 2, now this is analogous to metaphase in mitosis. Each chromosome is going to line up along the metaphase plate, and the kinetochore microtubules are going to attach to one of the 2 sister chromatids, one coming from each pole of the spindle and attaching to the kinetochore on each sister chromatid. Anaphase 2, as always, is going to disassemble those kinetochore microtubules at the kinetochore side, pulling those sister chromatids to opposite poles so that they can be separated during telophase and cytokinesis into separate nuclei and then eventually into separate cells as this cell pinches off by the formation of a cleavage furrow. Each of these 4 cells is going to contain one of these sister chromatids from one of these homologous chromosomes that resulted from meiosis 1. So at the end of meiosis, there are 4 daughter cells, each one with a haploid set of unreplicated chromosomes. So each one gets just 1 sister chromatid. And each daughter cell ends up being genetically distinct from the others and from the parent cell. And we'll see in just a moment why these, even though they look the same in this illustration, actually end up being different. This is due to this phenomenon called crossing over. So let's take a look at crossing over and synapsis, and this is going to occur during meiosis 1, during prophase, where the 2 homologous chromosomes are going to find each other and pair up. So after interphase, the sister chromatids are held together by proteins called cohesins. Each of these strings is one of the sister chromatids that was formed by replication, and these are held together by these cohesin proteins. So the blue ones, again, are the paternal chromosome, the 2 sister chromatids due to replication. The red ones are the maternal chromosome replicated to form sister chromatids. So between these 2 homologous chromosomes, a structure forms called the synaptonemal complex that holds the 2 homologous chromosomes together. So these are going to be lined up gene for gene in a very precise way using this synaptonemal complex. So what happens is the non-sister chromatids of these homologous chromosomes, the DNA is going to break at precisely corresponding positions. So when I say that they're broken at precisely corresponding positions, remember that these two homologous chromosomes have all the same genes in all the same order, so the exact same loci. So these two sister chromatids are going to be broken in places with exactly comparable DNA sequences. They're going to be broken and they're going to switch. So here we see the paternal homolog joining together with the maternal one in both of these cases. This is the crossing over. They're actually going to exchange genetic material. So during this period called synapsis, where the two homologous chromosomes are held together, the DNA breaks are repaired, and it's going to join DNA from one non-sister chromatid to the corresponding segment of another. This is that process that we just saw, where the paternal part of the chromosome is joined to the maternal one, the maternal to the paternal, and so they're just exchanging sections of that chromosome. So I think you can see that a little bit better in this illustration. Here we have the two sister chromatids, maternal and paternal, lining up gene by gene in exactly corresponding positions using the synaptonymal complex. This structure forms called a chiasma, where they're crossing over. So this is where the DNA is being broken. The blue one is being joined to the red chromosome. The red one is being joined to the blue chromosome, and so they actually switch. So this chromosome now has a blue section. This blue chromosome has a red section. This is called crossing over, and it's just the exchange of genetic information of part of those chromosomes between non-sister chromatids. And so what you end up with in the end is between these sister chromatids, there's going to be one that has not crossed over, the one that does not come in contact with the homolog. There will be one maternal that has crossed over that's going to have a little section of paternal DNA, and then one paternal that's intact that hasn't crossed over, and then another one that has crossed over and carries some of that maternal DNA. So each one of these four chromatids ends up being distinct from each other. So here we see this, a comparison between mitosis and meiosis. In mitosis, you've got a diploid parent cell. The DNA replicates to form sister chromatids. In prophase, during mitosis, those two homologous chromosomes do not associate with each other. In prophase 1 of meiosis, they do, and it's at this point that that crossing over takes place, and these chiasmata, that's a plural of chiasma, these little X-shaped structures that form during crossing over can actually be seen microscopically during prophase 1 in meiosis, when these two homologous chromosomes associate with each other, completely separate in mitosis. So again, in mitosis, each chromosome lines up along the metaphase plate. Remember, in meiosis 1, it's the pairs of homologous chromosomes that line up together to be pulled and separated during cell division. Mitosis results in two diploid daughter cells that are identical to each other. In meiosis, there's going to be another round of cell division in meiosis 2, resulting in 4 haploid gametes that each have distinct combinations of genes different from the original diploid parent cell. So now let's bring this full circle to what I was talking about at the very beginning of the web lecture, of why this is so important to the process of evolution by natural selection. So the genetic variation produced in these sexual life cycles contributes to the process of evolution. So the ultimate source of genetic variation is always mutations. These changes in an organism's DNA sequence is always the source of anything that's truly new in the population. So that's the original source of genetic diversity, and here we see a bunch of examples of mutations in fruit fly that cause variation from this wild type. So these are all separate mutations that have resulted in a novelty or something new in the population. So what mutations do is they create different versions of the same gene. So it's the same gene. It's going to have generally the same job in producing some kind of trait or character, but it's going to do it in a slightly different way, and it's going to be found at the same position or locus on the chromosome. So it's going to be a variation of the gene, and this is called an allele. So different alleles are found at the same locus. They're different versions of the same gene. And these are created ultimately by mutations. So what happens during sexual reproduction is that the combination of traits and characters is shuffled in every generation. So the reshuffling of different alleles during sexual reproduction produces the genetic variation that you see in a population. That genetic variation results in variation in traits. So the behavior of chromosomes during meiosis and fertilization is responsible for most of that reshuffling, that genetic variation that's going to cause different combinations of characters in individuals from generation to generation. So we've seen 3 different mechanisms that can contribute to that genetic variation. So let's just review them in the context of producing genetic variation. So we saw the independent assortment of chromosomes during meiosis, so that you can have different combinations of maternal and paternal chromosomes in each gamete. Crossing over, recombining the chromosomes coming from the father and the mother to combine the traits from the mother and the father into a single chromosome that gets passed down. And then finally, random fertilization, the fact that you have a large number of different gametes with different genetic information in them that are going to come together at random to form a zygote. Let's take a closer look at each of these. So first, independent assortment of chromosomes. During metaphase of meiosis 1, the homologous pairs of chromosomes are going to orient randomly. So that means you're not necessarily going to have all the paternal chromosomes on one side being pulled toward one pole and all the maternal on the other side. It's just as likely to have a paternal combined with a maternal and vice versa in the 2 daughter cells that result from meiosis. So if we look at these 2 possibilities of the different ways these can line up during meiosis 1, the number of combinations possible when chromosomes assort independently into gametes is 2 to the n, where n is the haploid number. So here we have a haploid number of 2. We have 2 different chromosomes. And so the 2 different ways these can line up is paternal with paternal and maternal with maternal, or paternal with maternal, and vice versa. So these are only 2 possibilities, and they're equally probable when you have a haploid number of 2. So either you're going to get, ultimately at the end of meiosis, from this possibility, you'll get gametes with both of the 2 paternal chromosomes in 2 of these gamete cells, and the 2 chromosomes coming from the mother in this other pair of gamete cells, or in possibility 2, you'll get paternal, maternal, paternal, maternal, maternal, paternal, maternal, paternal. 2 to the n in this case is 2 to the power of 2, or 4. So, for example, for humans with a haploid number of 23, there are more than 8 million 2 to the 23rd possible combinations of chromosomes that you can find in gametes. So really, 8 million different distinct combinations of genes in each gamete provides a huge amount of potential genetic variation going from parent to offspring. Crossing over increases that variability by recombining the genetic material from the paternal chromosome and the maternal chromosome. So any number of variations due to this recombination of chromosomes produced by crossing over can occur because these chiasmata can form anywhere along the length of the chromosome, and the number of times it does that can also vary. So the crossing over produces recombinant chromosomes, which combine DNA inherited from each parent. And this crossing over contributes to that genetic variation by combining DNA from 2 parents into a single chromosome. This can happen any number of times in any location along the chromosome. And in humans, happens an average of 1 to 3 crossover events per chromosome per gamete. So again, this is another huge source of variation. And then finally, random fertilization adds to that genetic variability because any sperm can fuse with any ovum. So you've got a population of eggs in the mother, a population of sperm in the father, each with these some 8 million possible different genetic combinations. So putting these together into the diploid organism at fertilization, in the case of humans, with 23 chromosomes, the fusion of two gametes, each with 8.4 million possible chromosome combinations from independent assortment, can produce a zygote with any of about 70 trillion possible diploid combinations. So each individual truly is, in a very real sense, unique in the genetic variability that they possess. So let's think again about the evolutionary significance of this variation. So remember that natural selection results in the accumulation of genetic variations that have been favored by the environment. They're going to increase in frequency in the population to produce a change over time in that trait in that population. So sexual reproduction contributes to the genetic variation in a population, which again originates ultimately from mutations. So the more variation you have in a population, the greater the likelihood that there's going to be a variant that has an advantage. Sexual reproduction that produces all of this variation is highly advantageous in a changing environment. The more variants you have, the better the chances are that one of those variants is going to be well suited to whatever change occurs in the environment. So animals that always reproduce asexually are very rare. This variation that's produced by sexual reproduction is such a huge evolutionary advantage that even species of animals that have been found to reproduce asexually have found ways of benefiting from taking on genetic variability. So the example that you saw in your book was the example of this deloid rotifer that reproduces asexually, but also can incorporate foreign DNA from other species, usually closely related species, into their cells and actually incorporate that DNA into their genome to keep this genetic variability in the population to be able to respond in an evolutionary way to changes in the environment. So let's bring this back to Charles Darwin and how he formulated this model of evolution by natural selection. So remember that he based it on all of these observations, and one of the observations was that offspring tended to resemble their parents, so the idea of heredity, heritable variation, and also that there was a huge amount of variation within populations. This is really the foundation of the theory, but remember that although Darwin observed the results of this heritability and variation, he knew nothing about the causes of it. So unbeknownst to him, at around the same time that he was formulating this model of evolution by natural selection, there was a Gregorian monk working in what is now the Czech Republic named Gregor Mendel, who was at that very time, unbeknownst to Darwin, discovering the secrets of heredity and inheritance. And so this is what we're going to be looking into in the next chapter, is how Mendel started to unlock the clues that were going to be the key to completing Darwin's theory of evolution by natural selection by adding this component of genetics to it.